Hello friend. So now after the clinical phase of the bioequivalence study, now we are moving to the bioanalytical process of the bioequivalence study. So this is very important and critical process in the bioequivalence study. Anything goes wrong in the bioanalytical process, the entire study goes waste. So we have given as much information in this particular module. Relatively, this is one of the big module in the entire course. So hence request you to keep patience to complete this entire module at one stretch, and also keep all the informations, go through all the informations and understand as much as possible. Before going into the bioanalytical process, I would like to emphasize on some of the definitions of the basic science. First, the chromatography. Chromatography is a process for separating components of a mixture. To get the process started, the mixture is dissolved in a substance called the mobile phase, which carries it through a second substance called the stationary phase. So chromatography will have two different phases. One is the stationary phase and the mobile phase. And the compound in a mixture form, we need to separate, which will be there in the mobile phase, and by some mechanism, by using the stationary phase, the compounds will be separated. The mechanism generally partition coefficient or adsorption or it's any other. Then the chromatographic column, which is a device used in the chromatography for the separation of chemical compound. A chromatographic column contains the stationary phase, allowing the mobile phase to pass through it. Chromatographic columns of different types are used in both gas and liquid chromatography. So the chromatography column basically contains the stationary phase in the form of silica gel or something else, and the mobile phase will be going through the column. Then once the compounds are passed through the chromatographic column, then the detector comes into the picture. A chromatography detector is a device used in gas chromatography or liquid chromatography to detect components of the mixture being eluted of the chromatographic column. So the detector basically detects the compounds and help to quantify the compound in terms of the response. So this is very important device which is present in the chromatographic instrument. The most common liquid chromatography detectors are the ultraviolet detector, fluorescent detectors, and the refractometer, RI detector, using the refractive index. Apart from that, there are mass spectrophotometers also used as the detector in the chromatographic system. Now we move into the bioanalytical. What is bioanalytical means? The bioanalytical means identification and analysis or estimation or quantification of drugs, chemicals, metabolites, proteins, peptides, etc. in any biological matrix like blood, plasma, serum, urine, other biological fluids or tissues. So it is a way of identifying and estimating the compounds in a biological matrix. In bioequivalence analysis, we estimate the drug in blood, plasma, serum using the bioanalytical methods. So the bioanalytical process is comprising separating the drug from the biological matrix, that is extraction of the drug, and then quantification of the extracted drug by the chromatography. There are two processes which is comprised in the bioanalytical process in the bioequivalence studies. Bioanalysis employed in various activities of pharmaceutical development, including preclinical, clinical studies, toxicity studies, etc. So apart from the bioequivalence studies, these bioanalytical process are being used in preclinical studies where the animal pharmacokinetics are established. Toxicity studies, drugs will be measured from the organs for the toxicity. So the applications of bioanalysis is wide open, and here we are focusing on only the bioequivalence studies. Ideally, bioanalytical methods should be developed for specific drug before initiation of the clinical phase of the study, because the bioanalytical procedure defined the blood sample handling procedure during the clinical phase of the study. So we have to ensure that the bioanalytical method is developed and validated before start of the clinical phase. In this slide, we are seeing the bioanalysis purpose in the bioequivalence studies in a detailed manner. Once the drug is given into the human body and goes into the gastrointestinal tract, it gets absorbed, distributed, metabolized, and excreted. Except the absorption, except the intravenous route, there is no absorption for the intravenous route, but other dosage form there will be absorption. Then the drugs are metabolized and becomes metabolites, sometimes active metabolites before the excretion. The metabolites also can provide some action, pharmacological action. So in the bioequivalence study, we measure the drug concentration in the systemic or blood circulation with respect to time. For this, we collect blood samples at different time points. To measure the drug and metabolites in the collected blood, plasma or serum samples, bioanalytical methods are employed. Upon clinical phase of the study completion, the blood samples are received at the bioanalytical lab and stored till the analysis. So this is how the bioanalytical purpose of the in the bioequivalence studies is described. Now we are going to talk about the instrumentation adopted for the bioanalysis. So in the older days, HPLCs are the most commonly used instrument in the bioanalysis. That is 30 years back. HPLC are the widely used instruments. Nowadays, the LCMS, liquid chromatography mass spectrophotometer, is the most commonly adopted instrument for the bioanalysis for because of its versatility for the purpose. The liquid chromatography part can be HPLC or UPLC, ultra pressure or high pressure liquid chromatography used for the liquid chromatography purpose in case of the LCMS. Then the MS will be the mass detector, it will be the backend for the instrument. So the chromatography is the important technique in bioanalysis, and liquid chromatography is the most adopted approach in case of the bioequivalence analysis, because we receive different kind of drugs which is cannot be used for the gas chromatography because of the adaptability or the suitability of the drug compounds for the method. So mostly we go for the liquid chromatography, that is through the LCMS. The chromatography separate the drug from the given samples and detected through detectors like mass detector or UV-PDA in case of it is a HPLC. So these entire instrumentations are very critical. The working functioning nature of the instrument is very critical so that you are producing accurate data out of the analysis. So for that, the instrument needs to be qualified. Once you buy the instrument and keep it in CRO, it should be qualified. The qualification will be installation qualification, operational qualification, and performance qualification. And every 6 months or 1 year, the instruments are calibrated so that it will give you a accurate results. Then the software is used for the chromatographic construction as well as the estimation of the compound should be validated for the computer system validation, and it should be compliant with the guidelines prescribed in 21 CFR Part 11, which is the US FDA guidelines. So this is prescribed for the computer systems. So it has to comply. There should be audit trials. So each and everything you deal in the software will be recorded. Any changes, anything you add, delete, everything will be recorded and it will be captured in the audit trials. So this is how the instrumentation of the bioanalysis is being described for the bioequivalence studies. So this particular slide talks about the instrumentation of LCMS, how it is looks like, and then how a liquid chromatography separation happens, and what is front and back end. So here you can see the mobile phase container, and from this the mobile phase will be going into the instrument, and this is solvent mobile phase, which will be in the A and B bottle will have different gradients of the mobile phase. The pressure will be created in this portion, and the columns will be kept here, and then once the sample will be injected through the instrument, the mobile phase will carry the sample and goes through the column where the compounds will be separated, and then it will be going for the detection through the mass spectrophotometer. Then the entire data will be processed through the computer software, and it will provide you the chromatogram where you can see the quantification of the results. So this is how the entire LCMS setup. This is the LC part and this is the mass part, and in between the column comes, and this entire setup are kept in the bioequivalence CRO setup. And this is original mass spectrophotometers from the SciEx, and this is how the LC separation will give you chromatograms like this different. If it is a mixture of the compound, this is how the compound will be separated, and it goes through the mass detector, and it will give based on the ions. It will be the selected ion will give the quantification of the particular drug. This slide describes a reverse phase high pressure liquid chromatography chromatogram where the mixture of compounds are injected and the injected compounds are separated in a chromatogram based on the retention time. So this particular compound, the first compound, is eluted in the retention time of in between 5 to 7.5 minutes. After that, the second compound eluted at around after the 7.5 minutes, and finally you have around 13.3 minutes, you have another peak is eluted. So depends on the retention time, the compounds are eluted and well separated in this particular chromatograph. This is the samples of mass spectra where the different ions are detected with the mass spectra, and one of the ions from the drug molecule will be used to determine the quantification. So here you can see the internal standard peaks and other mass ions of the particular drug, but specific mass will be selected. M by Z will be selected for the quantification of the particular drug. This is the typical mass spectra you are seeing in this particular slide. Now we move to the bioanalytical method development. We have seen sufficient information about the instrumentation, and now the chromatography development we are going to see. For this purpose, we procure the pure drug samples, that is called working standard or the active pharmaceutical ingredient qualified for the purpose of the bioanalysis you can use. So pure drug samples, generally referred as working as standard, will be procured for the bioanalytical method development purpose. Then the aqueous solution of the drug are injected into the liquid chromatography mass spectrophotometer, or non-aqueous solutions like methanol solutions, methanolic water, many combination of these solvents can be used as a diluent for the purpose of dissolving the drug and inject into the LCMS. Then the chromatography method will be developed for the separation of the drug at a retention time. So that first will be developed, the chromatographic condition will be established for the bioanalytical method. So the retention time is specific for specific drug in the chromatography. In case of two drugs are there, some other compounds are there in the matrix, that will be separated out, and at a specific retention time the particular drug will be eluted. So the RT is specific to the drug will be very critical during the chromatography development. Then after the initial chromatography development, the mass detectors will be used to for the quantification. So specific mass of the drug will be selected for the particular method. So at the specific mass M by Z, the quantification of the drug will be detected and quantified. The chromatography checked for the method is specific to the drug. That means if you inject some other things, it will be eluted at some other RT, and the drug only will be eluted. That will be the specificity will be assessed when you use the drug, spike it into the matrix and you inject, you will get the drug. If you inject only the matrix, you won't get anything. Or the placebo, if you are injecting, you won't get anything. So that is called the specificity of the drug. If you change the concentration of the drug, for example, 10 microgram, 20 microgram, 30 microgram, the response, what you are getting, the peak height or the peak area has to increase. From the 10 to 30, it has to be an increasing trend. That is called the response to the concentration, and it should be linear. So these are all the parameters initially checked during the chromatography method development. In general, during the bioanalytical method development, the internal standards will be used. So in olden days, there is no internal standard. So, but when the mass spectrophotometers came into the picture, along with the liquid chromatography, LCMS came into the picture, the internal standard usage came into the picture. Why internal standards are used during the bioanalytical method? Internal standards are widely used in mass spectrophotometry, not only to correct for the sample preparation variations during extraction and chemical derivatization, but also to compensate for variability in signal intensity due to ion suppression caused by matrix components that may influence the efficiency of ionization. It means in case if you use internal standard, for example, you take the drug in the biological matrix, spike some concentration, and also you spike the internal standard. So both the things are spiked. Internal standard concentration will be always constant. You spike in a constant concentration in all the samples. So when you then you extract the samples, so the extraction efficiency will be applicable both the drug as well as the internal standard. Then it will be go for the separation and the quantification. During quantification, the ion suppression can be caused by the matrix components, though that will have the impact on both the internal standard as well as the drug component. So whatever the response you got the internal standard, it is directly proportional or it can be similar to the drug response. So based on this internal standard peak area and the drug concentration peak area, there will be a ratio will be calculated, and that ratio will be used for the quantification. So this is how the variations happens during the extraction, during the ion suppression, So this all will be eliminated through the internal standard usage for the appropriate quantification of the drug. So the main purpose of utilizing internal standard is to improve the accuracy and precision of the quantitation as well as the robustness of bioanalytical methods. So the internal standards are recommended to be similar in structure and properties, and in practice deuterated the isotope of the drug molecules. In case of paracetamol, then we take for the deuterated paracetamol as the internal standard, where the physical nature of the drug is remains same and the chemical nature of the drug is not the same. So this isotope based internal standard used, it possesses different mass, so the physical nature of the things are different, so the mass will be different, but the chemical nature of the compound will remain same, so that it will behave similarly. So internal standards will be used in single level concentration. As I said earlier, it will be spiked in the same constant concentration. This particular chromatogram obtained from the mass spectra so shows the response by the internal standard as well as the drug histamine, for example. And this particular plot, this one and this one, showing the internal standard variations by injections. So ideal internal standard variation should be like this. This should be the ideal things required. In case if you found the variations like this, then the method is not a good one. So the variations, injection to injection variation of the internal standard response by peak area should not vary much, and it should be probably 10 to 15% variation is acceptable one. This is very highly variable and it should not be accepted. Now we move to the extraction method development for a bioanalytical method development. So basically the drug is present in the blood matrix. Then the blood contains proteins and complex biological material which can interfere with the bioanalytical method. Hence, separating drugs from the proteins and the biological materials is critical. So the different extraction techniques are adopted to separate drug from the matrix, that we are going to see in the coming slides. The generally adopted extraction methods are protein precipitation, liquid-liquid extraction, and solid phase extraction. So the protein precipitation is simply adding organic solvents to the biological matrix, which precipitates the protein and separate the drug. So we do the protein precipitation by adding the organic solvent, filter it and purify as much as possible the drug layer and take for the analysis. Then the liquid-liquid interaction. So when you add it is based on the partition coefficient, you know the solubility of the drug. Based on that, you go for the organic solvent extraction of the drug from the biological matrix using the liquid-liquid extraction using separating funnels and some other methods. Then the solid phase extraction method which comes into the picture. This is expensive and most adopted method which gives more extraction efficiency than the other two methods. The first one, protein precipitation, is the simplest method, and liquid-liquid extraction is the tedious method but cost-effective method. But the solid phase extraction is some kind of mini columns and cartridges where I explained to you how the HPLC functions. Like that HPLC, that means you are injecting the compound mixtures through the columns and separating out through the elution. Like that, it is in the miniature columns will be used to separate the drug from the biological matrix. The column cannot be reused and it will be thrown out. So it is a single time use. These kind of mini column cartridges will be used to separate the drugs from the biological matrix. This is called solid phase extraction. So in this particular slide, we are going to see schematically how these extraction techniques are functioning and how it is useful to separate the compounds. First is the protein precipitation. Here is the biological matrix where the drugs present. Then we have the insoluble complex, and then you are adding the organic solvent with this particular vial. Then you will get the protein all getting precipitated here, and there will be a supernatant. Then the supernatant will be taken separately by centrifuging, and then the supernatant will be taken for the reconstitution and used for the injection purpose. So the precipitate will be separated, then either the supernatant will be taken separately for the analysis, or it can be reconstituted into the other solvent and taken for the analysis. This is how the protein precipitation techniques are used. And this one is the liquid-liquid extraction, where you have the compound present in the biological matrix. Then you add the organic solvent and do the extraction by mixing the compound present in the biological matrix, eluted or dissolved into this organic solvent. Then you separate this solvent by separating funnel, and you get the drug into this organic solvent, which will be used for the further chromatography. Now you have this solid phase technology, where you have the cartridges column-like structure where you load the sample through this, and you have the unwanted component are captured by the column cartridges, and only the compound you need will be eluted through the column, and which will be collected, and further it will be taken for the chromatographic analysis. So this is how the entire extraction techniques are followed in the bioanalytical process. Now, the bioanalytical method is developed. So now the chromatography and extraction methods are finalized. Now the calibration curve method will be used for the quantification. What is the calibration curve? Calibration curve is the drug concentration versus response curve established in a range of concentration, and a linear regression of the curve will help to calculate unknown concentration based on the response received for unknown samples. So this we will see in the upcoming slide. So we discussed about the calibration curve. So there will be a range of concentration will be used to construct a calibration curve. So there will be the lowest concentration in the calibration curve is called as lower limit of quantification. Then the highest concentration in the calibration curve is called as upper limit of quantification. That is ULOQ. The lowest concentration at which the drug peak can be detected in a chromatogram is called as limit of detection, and always it is lower than the limit of quantification. The LOD will be always lower than the LOQ. With the LOD, you can just detect the compound. With the LOQ, you can properly quantify the compound at the minimum level. Calibration curve are constructed based on samples of drug spiked blood samples at various concentrations. These are blank blood samples which are used for the method development purpose. These blood samples, you would spike the drug at various concentrations, and you construct the calibration curve by doing the analysis. In this particular slide, we are seeing the typical calibration curves. So here is a calibration curve. This is the LLOQ, lower limit of quantification. This is the ULOQ, upper limit of quantification. And below this, you can have the LOD. And this is the unknown concentration, how it is being extrapolated using this linear regression of the calibration curve. This is described here. So this is the unknown concentration, and based on the response you get for the unknown peak, the peak of the compound for which you don't know the concentration, the response you know, based on the response you extrapolate the concentration of the drug. This is how it is being calculated using the calibration curve. This is also a typical calibration curve, where the points are scattered all over the places. It's not a great curve, and here you can see the how the working range is defined in using the calibration curve. So this is the LOD, this is the LOQ, and this is the upper limit of quantification. So from here to here we call it as a working range. So you have to bring the concentration of the drug by dilution. In case it is high concentration, by dilution you have to bring to this working concentration range, so that you can easily quantify the compound. Suppose some samples giving very high concentration, this is not a validated range. If for example here your concentration is there, it is above the working range, means you have to dilute these samples, again you have to analyze it. That is how this working range is useful for the bioanalytical quantification of the drugs. In case of bioanalytical methods, you will always use quality control samples. So the quality control samples are very important because it will be used for the run acceptability. So quality control samples are prepared by spiking the known concentration of the drug in the blood samples. The concentration of the drug will be in the range of the calibration curve. It should be within the working range, what we discussed in the earlier slide. So there are at least 3 to 4 quality control samples at different concentration, right from the lower limit to ULOQ will be prepared, and each and every analytical run these quality control samples will be processed and also will be injected along with the samples and give the responses. So the run acceptability of the bioanalytical method will be based on the response or the concentration, back calculated concentration provided by the quality control samples, and it should be within 15% or 20% as defined in the guidelines. If it is more than that, the run will not be accepted and it will be a failed run, and you have not processed the samples properly, or you have done some mistake during the analysis. So this is how the importance of the quality control samples comes into the picture when you do the bioanalysis. And these quality control samples are interspersed between the samples. If you have 20 samples to analysis, you have to put after every 5 samples one quality control samples you have to put. That is how the quality control samples are used for the bioequivalence studies. In continuation to the previous slide, the quality control samples, there can be LQC, which covers the lower concentration, MQC, the middle concentration, and the HQC, high concentration, and sometimes additional QCs may be included as and when required for the bioanalytical methods. And the QC samples, as I said earlier, which is the results of the QC samples is critical for the analytical run acceptability. And always for every run, you should use a freshly prepared QC samples from the stock already prepared for during the bioanalysis. So this particular slide talks about how the QC samples are constructed across the working range or the calibration range of the particular bioanalytical method. Here is the high QC, here is the middle QC, and here is the lowest QC being used. Sometimes you have additional QCs in between this range. So this is how the QC samples are defined during the bioanalysis process. So now you have the method is already developed and the basic concept of the bioanalytical method is understood. Now, whether the method is perfectly all right, whether it is working all kind of parameters or not, how we can prove the method. So for that we have to do the validations. But before doing the validation of the method, we need to verify the method during the development stage itself, because validation is a huge GMP process or GLP process, and if it fails, you have to go back and do it can be a redevelopment or something. So before going to the validation, you need to verify the method for some parameters. So those parameters are explained in this slide as well as the upcoming slides. So first parameter is the specificity and the selectivity of the method. What is specificity and selectivity? It should be evaluated to assess the interference at the retention time of the analyte and the internal standards with predetermined method conditions. If the single method assesses one or more two analytes simultaneously, the interference should be evaluated separately for each analyte individually. So the selectivity and specificity has to be established for each and every analyte to be determined in the method. The selectivity of the method is routinely demonstrated by analyzing blank samples of the appropriate biological matrix from the multiple sources. So you have to collect the blood samples without the particular drug of analyte, and then you have to use as a blank for the specificity experiments, specificity as well as the selectivity experiments. So the different sources of the blank samples will be injected, and the spiked samples also injected, where you will get the peak only in the spiked samples, not in the blank samples. This is how the selectivity of the method will be established. Then depending on the intended use of the assay, the impact of hemolyzed samples, lipemic samples, or samples from special population can be included in the selectivity assessment. So what is the difference between the selectivity and the specificity? Specificity about the method where the blank sample doesn't show the peak and the drug spiked sample showing the peak. Then the selectivity will be different blood samples. The blood sample during the study you may get hemolyzed, you may get more fatty blood samples, or you may get the aged people samples or the lower age people samples. So all these samples show similar response. That is the objective. So the selectivity of the method has to be proved by using different sources of the blood samples from different sources like hemolyzed samples, lipemic samples, special populations, etc. And the matrix effect has to be proved for the selectivity. And the specificity, as I explained, it has to show only the peak for the spiked samples, not for the blank samples. So this is all about specificity and the selectivity of the method. Next is the sensitivity of the method. So the sensitivity of the method is defined by the limit of quantification. So the up to this limit of quantification you can use the method. Below that you cannot use. That is called sensitivity. So the lowest standard LOQ should be accepted as the limit of quantification of the method. This test should be performed. That is the sensitivity test should be performed to prove the reproducibility of the samples at the limit of quantification level. So sensitivity should be evaluated by using at least 5 replicates of the samples at the LOQ. The samples used for this evaluation should be independent of the calibration standard. So we have to prepare the LOQ samples and inject multiple times and see the reproducibility, and there should be a acceptance criteria to it should be within the precision of the LOQ values should be within this much percentage, 10% or 15% has to be established. So then the sensitivity of the method has been proven. Next comes to the accuracy and the precision of the method. So what is accuracy? Sometimes it is called as the recovery. So the accuracy and recovery is the different terminology used to denote the same thing in the bioanalytical method. So the accuracy and precision. Accuracy is the degree of the closeness of the determined value to the nominal or known true value under prescribed conditions. So you developed a method. Now you are injecting a spiked samples at the ULOQ value of 100 nanogram per ml. So now you are injecting that and you are getting the results. That is the back calculated value around 89 nanogram per ml. So the back calculated value gave you 89 and you spiked theoretically 100. So now what is the accuracy? Your accuracy is 89%. Okay. So this accuracy, there will be limit for accuracy where it's generally in bioanalytical it is plus or minus 15% for the different quality control samples like LOQ. LOQ it will be more where it can be extended up to 20%, but in case of the other quality control like middle QC and the high QC, it will be 15%. So 89% is acceptable, but as you give more accuracy, it will be a good method. So the variation will be less and your robustness of the method is good. Then it comes to the precision. Precision is the closeness of agreement among a series of measurements obtained from multiple sampling of the same homogeneous sample under prescribed conditions. This means you are injecting the same samples 6 times or 3 times or 12 times and the number of times you are getting different results. So this variation should be within some kind of limit. So you generally the precision should be less than 15%. So when it is comes to the less than 15%, the variation will be less than 15% among 3 times or 6 times injections of the same samples, then your precision is good and it is acceptable. So the variation amongst the results are called the precision and the closeness of the results to the actual value or the nominal value called accuracy. Hope this is understandable. When we see some examples, it is more understandable. So the accuracy and precision should be assessed by analyzing at least 3 batches, including both intra and inter-day runs. You can do 2 batches in one day and another batches another day so that you can assess the variation between the inter-day and intra-day. So intra-day 2 batches means intra-day. Inter-day means one batch you are doing in another day. So you can assess this intra-batch variation and inter-batch variations by doing 3 batches of accuracy and precision. Both within and between run precision also can be established. So each validation batch must consist of at least 6 to 8 non-zero calibration standards, one standard blank and one standard zero, and 6 replicates of quality control standards at each concentration, QC concentration, that is LOQ, LOQQC, limit of quantification, then low QC, middle QC, and high QC. There are 4 concentration you should be used within the working range, that is the calibration curve range. Then there will be 6 replicates of this concentration and there will be a standard zero, there will be standard blank, and this entire set of samples will be injected in one batch. That is the accuracy and precision batch, P&A batch, what we call, and it will give you the results for the accuracy and precision. So this particular slide giving more detailing and more understanding about the precision and accuracy of the particular data. So this first we'll go to the table. We have the concentration of different QCs 6 times injected. So this is the 6 values you get for the low QC, that is the lower limit of quantification. Then you have the LQC and then the middle QC and the high QC. Now you have the mean value. So this value is nothing but your accuracy of the given. These all values are the accuracy values, and this mean value is the mean accuracy. And in terms of percentage, this is the nominal value against the obtained value. So nominal value is what is the actually theoretically you added, and this is what you have got the back calculated concentration. In terms of percentage, it is 100%. This is 99%, this is 97%, and this is 104%. So when you call this all comes under the plus or minus 15%, so it is acceptable, and this should be plus or minus within plus or minus 20%. This is also acceptable. So the accuracy percentage is calculated against the nominal concentration. This is what you spiked, and this is what you got. So in terms of percentage, the accuracy is calculated, and then the precision. Here it is 6.4%, and this is 10%, 9.2%, 9.3%. Precision is calculated by using the standard deviation. So here 6 values, how it is varying. So this standard deviation divided by mean into 100 will give you the CV. So this is nothing but the precision. So standard deviation divided by mean into 100, which will give you the precision value. And this is how the accuracy and precision is calculated from the precision and accuracy batch data, P&A batch data. And how the accuracy and precision is useful for the method determination or the conclusion of the method. So here, if you should get this ideally these kind of results where your method is more accurate and more precise. Everything is inside the bullseye. So your different values are very close to each other, and the mean value is the yellow mark is the mean your spiked value. So you are in the spiked value range and all the values are within the spiked value range. So your method is more accurate and more precise. Here it is accurate, it is close to the bullseye where the nominal concentration, but it is varying at the different range. This value may be higher side, this value will be the more lower side. So this is not very precise. And this one is not accurate, neither accurate nor precise. And this one is not accurate, is not to the near to the yellow line, but all the values falls close to each other, so it is precise. So this all these three things are not good for the method, and this is the only good method based on the accuracy and precision data. Hope this slide explains you how the precision and accuracy is determined for during the bioanalytical method. Now we are coming to the important parameter recovery. So recovery is sometimes confused between the accuracy and the recovery, but recovery actually explained here. Recovery can be determined by comparing the detector response of the analyte or the IS from an extracted sample to the detector response of the analyte from an unextracted sample, representing the 100% recovery. So what is unextracted sample? So you can say that aqueous or a neat solution, whether it is whatever the solvent, you are adding the drug and making the solution which is having 100% recovery. That means the neat solution of the drug is 100%, or a blank plasma sample where you add the neat drug solution which is can be called as a 100% recovery. So the recovery will be evaluated at low, middle and high QC concentration, and the average recovery of the analyte and the internal standard at three levels could be considered as the recovery. So the recovery should be always, generally we try to achieve with the extraction method very high, and sometimes if it is low, and it should be consistent. It should not be vary. So that is more important in case of the biological matrices. Now come to the stability evaluation, which is very critical for the bioanalytical method. So this stability will determine how much time you can keep the drug in the solution form, neat solution stability, then how much time you can store the blood samples, at what temperature you can store. All those things are determined by the stability evaluation. Drug stability is a function of the chemical properties of the analyte and storage conditions. Matrix are the solutions in which it is stored, and the container system. The stability tests should reflect the situations likely to be encountered during the actual subject sample handling and the analysis. The stability of the analyte in the matrix during the collection and storage of the sample should be assessed. Various stability like short term, long term, bench top. Bench top means whatever sample you prepare and keep it in on the particular day. Then the auto sampler freeze the how many times you take the blood samples, return to the freezer and again you are taking, then the solution stability, matrix stability will be evaluated. So all these data is very critical for the sample handling procedure, and the storage is determined by the chemical property of the drug and which matrix you are storing the drug, whether it is a pure neat solution or the blood samples or whatever it is, which container system, glass container or some other things. This all can impact the stability conditions, also the storage condition. Then it comes to the another couple of parameters where the dilution integrity. So dilution of the study samples will be performed when the obtained high concentration or when the obtained concentration is exceeding the upper limit of quantification, or when there is a less sample availability compared to the method requirement. So as I told you, there are the working range, and beyond the working range, if the concentration is obtained for some samples, then you have to dilute the samples and analyze. Or if you have a you need is zero point 0.5 ml of the plasma for the analysis, but you have only 0.1 ml. In that case also, you can dilute the sample and analysis. For that, the integrity of the samples after the dilution need to be proven during the verification stage. So, for example, you spike the 100 nanogram per ml in the blood sample, blood matrix, and you extract the samples and you inject it, and then you dilute that 2 times or 10 times and inject. So the recovery, what you are getting, the accuracy of the samples, what you are back calculated condition of the diluted samples, when you diluted 2 times, you should get 50 nanogram per ml. You diluted 10 times, you get 10 nanogram per ml or close to that, within plus or minus 15%. Then your integrity is passed. Then comes to the carryover. The carryover test will be performed to observe any carryover from the instrument from one sample to the next sample. So the injections are happening next to next. So to evaluate the carryover, a blank sample will be placed next to the ULOQ standard in the sequence. Calculate the absolute peak response as percentage of the LOQ standard. So you are going to inject the largest concentration, then followed by a blank sample, and there should not be any drug concentration in the blank sample for when it is injected next to the highest concentration. Hence, it proves that there is no carryover of the drug during the subsequent injections. So if the carryovers are there, then probably you have to give some kind of time for running of the mobile page or something. So some alternate approaches will be needed. So once the methods are verified after the development, then once it is acceptable, then you can go for the bioanalytical method validation as a using the method validation protocol. The difference between verification and validation is that in validation you have predefined a criteria. For example, specificity, you should not get the peak in the blank, and then the precision, you have the 15% for the high concentration, middle concentration, etc. For LOQ 20%. Accuracy, you have same kind of a 15% criteria. Then LOD, LOQ has to be, there should be some criteria for the sensitivity of the method. Stability, you should get within 10 to 15% from the against the neat solution freshly prepared. Then the carryover, dilution, and integrity also show the similar kind of criteria. And the matrix effects, robustness when you change the column in the sense, if you are change the flow slightly the flow rate or slightly the column dimension, then you should get the results within the acceptance criteria plus or minus 15%. So the accuracy and precision, most of the cases, accuracy precision should be proven by changing the different conditions, different storage period, or the different anticoagulant. If you want to use the anticoagulant K2, K3, it also show the similar results. So this all should be validated by a by running all the parameters with a predefined acceptance criteria called method validation, and it should be done under the quality management system, and it should be reported as per the GLP procedure. Now the bioanalytical method is developed. We understood the instrumentation first, the chromatographic principles first, then the instrumentation, then the method development, then the verification and the validation. So all these then sequentially done. Then finally the study samples will be analyzed using the developed bioanalytical method. The stored samples analyzed using validated bioanalytical method. Each analytical run should be accepted based on the acceptance criteria for calibration curves and quality control samples. So CC also should pass with the predefined criteria, and quality control samples, as I said, it should also pass. Then only the subject sample analysis, each analytical run will be accepted during the study analysis. If a run failed, repeat injections or repeat analysis will be carried out with the predefined criteria. You cannot do the repeat injections or repeat analysis with the appropriate justification or appropriate documentation. Then at the end of the all analytical runs, incurred sample reanalysis, some selected samples at different levels, different time points will be carried out for to confirm the obtained results and the method. So this is the entire process of the study sample analysis. With this incurred sample analysis and all the analytical runs for the study samples, the bioanalysis will be getting completed. So after the completion of the study samples, the concentration data will be obtained and will be reviewed by the quality control department and the quality assurance department. Then finally, the quality assurance data will be used for the pharmacokinetic and statistical analysis Parallelly, the bioanalytical report will be prepared by the bioanalytical investigators and the staff, and all the information related to the analytical method, sample analysis, which date you received, which date you started the analysis, what kind of results you have got, and each and every analytical run, CC tables, QC tables, all are presented in the bioanalytical report along with the incurred sample analysis data. Then the results are tabulated, all the QC results, that means quality control sample results will be tabulated to understand the analysis throughout the study. There is a global QC results, individual run QC results, all will be reviewed by the reviewers as well as the investigators. So the report will be containing the method validation report and the standard testing procedure used for the method as the annexures. So with this, the bioanalytical method development, validations, and the study sample analysis, reporting the data gets completed, and the bioanalytical phase of the bioequivalence study gets completed. With this module, we have seen the bioanalytical phase of the study in a detailed manner. We completed the clinical phase of the study and the bioanalytical phase of the study. And going forward in the upcoming module, we will see that how the pharmacokinetic and statistical analysis are being carried out, how it will be reported. Thank you.